Theory of Phage and Bacterium. A presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. Elementary unit of biology. Hitherto, we regarded cell as elementary unit of biology. Cells come from cells, as we learned from Rudolf Virchow. In his language, it is omnis cellula as cellula. Yet, new cells never remain alone. They always join a family, and therefore cell family is the elementary unit of biology. So we are talking about Escherichia virus type, which is the name of the bacteriophage. And you see here three representations of this event. First of all, we see here the bacteriophage injecting or infecting a bacterium, and here the bacterium have a nucleus with DNA, and it is a prokaryote. But I deal with this interaction between virus and cell in several presentations and generally use this kind of representation. And recently I also used another kind of description, which is virus-provirus process. So I shall continue applying this kind of representation to the bacteriophage. So we have here the virus here. Remember, it's a bacteriophage attaching to a receptor, and this cell is a bacterium, a prokaryote. So we start with this representation and realize that this bacteriophage attaches to a receptor and activates a provirus, which is called prophage, and this prophage or provirus controls whatever happens in the infected bacterium or cell, and the provirus is also responsible for creating new virions. So let's see some definition. Provirus or prophage is a set of cell genes with three functions. It stores the self of a cell, it controls who may infect the cell, and controls virus production. And the self is a set of provirus genes unique to each cell. So here you have it. The phage infects the bacterium and activates the provirus, which is a prophage, and the prophage is then responsible for creating virions. And here you have the definition of virus, which is here the bacteriophage. Virus is a cell messenger. Virus is created by provirus. Virus stores self of the cell in its genome, and virus infects only cells with the same self. And again, self is a set of provirus genes unique to the cell. So let's look at two kinds of virus infection. So this is virus which infects a cell, or here we are talking about phage which infects the prokaryote. And so we have this relationship, and since the virus carries the same self, the virus is attached and it enters the cell, and the virus triggers the formation of provirus or prophage, etc. But here you have another virus, which is also a phage, which is not, does not belong to the virus like which I call one of us, and it is regarded as a foreigner, and therefore the provirus, which knows who is one of us, does not let the foreigner infect, and nothing happens. So here we have a virus infection which is blocked. And this is the whole idea of the viruses, that they infect only cells in families. So here you have an example. Here you have a cell family, and we are talking about prokaryotes, bacteria, etc. And this family starts from a cell which is a clone founder. And this clone founder creates the family which we are talking about. And the main problem of this family to know who is one of them. And so cells of a family carry a unique self stored in their genome, and the virus is the messenger of the family self, and it infects only members of the families, and therefore the family knows who is one of them. So, virus kinetics. Virus dies during infection, which means here you have the virus, attaches to a receptor, and then there is an uncoating, and leave only the genome of the virus in the cell. And this uncoating causes death of the virus, because uncoated virus is dead. So virus is born. After uncoating, the provirus or prophage is activated, and it creates virions, and this is where the virus is born. Virus is born in a provirus. And now about the cell. Cell is born during mitosis, and cell dies following virus infection, which is called lysis. So here you have the kinetics, everything changes, and I describe it in several presentations. So conclusion, virus is a cell messenger created by the cell. In our, we say that phage is a cell messenger created by the cell. No cell means no virus. More to come. Enjoy.